right, so let's get started. Right, so it's a real honor to be here today to talk to you all about what I think is an important vision, a collective ambition that kind of grew out of experiences within the BPI and a few other institutions on campus. And I'm going to talk to you about microbial cell systems for sustainable living. But before I do that, I just want to acknowledge the fact that everything scientifically that I'm going to talk about today is not done in a vacuum. It's done through collaborations with amazing people, both here at UBC, within my own lab, and across departments and units, as well as internationally with folks all over the world. We can only solve big problems together, and hopefully I'll convince you of that even further at the end of this talk. I also want to acknowledge the various funding sources that we require in order to make this work happen. And that's everything from our provincial and federal funding agencies like Genome BC and Genome Canada and NSERC, all the way through to some support through the Department of Energy in the United States. So we're going to walk a narrative path that has three specific stopping points, if you will, to highlight this, this journey. The first is I'm going to talk about living in a microbial world. And then I'm going to talk about harnessing the power of microbial communities that exist in the world. And then finally, I'm going to talk specifically about how do we cultivate an innovation ecosystem around this idea of biotechnology innovation related to this microbial world specifically. So we do live on a world that is dominated by prokaryotic microorganisms. There are an estimated 10 to the 30th power bacteria and archaea on the planet. And I just want to pause for a moment because that's a big number. And I don't know why I'm going forward. Okay. 10 to the 30th bacteria and archaea at any given time. That is more than the number of stars in the known universe at 10 to the 22. It is more than the number of synapses in our individual brains. And it is more than the number of collective synapses in all of humanity combined. So there is an immense possibility encoded in the biological information within the genomes of that vast microbiota. So if we do a back-of-the-envelope calculation, there are approximately 10 to the 33 genes encoded in that 10 to the 30 bacterial cells. That represents about 10 to the 26 gigabytes of actual sequence information. So if we think about this planetary system as an information encoding engine, the prokaryotes are the dominant information component of the planetary operating system. And that operating system collectively drives the biogeochemical cycles that create and sustain life on the planet. And it has been doing this for over 3.5 billion years of evolution. So this is a wiring diagram of sort of key transformations that define the evolution of the atmosphere, that define nutrient cycling. And microorganisms are the key players in all of this, although we are trying to contend with them in a few of these areas ourselves. Penny Chisholm, professor at MIT, wrote in 2000, the regulation of the pools and fluxes in biogeochemical cycles have their origins in the genetic inventory of individual microbes, and the regulation of these genes within the organism is determined by the environment. As such, one can look at the microbial food web, this is important, the microbial food web, as a collection of genomes whose expression and replication is coordinated through complex feedback loops at the organismal population. Here she writes ecosystem level, I will substitute community level of biological organization. And they do this through a nested hierarchy of metabolic interactions. Individual cellular agents, individuals may encode metabolic processes, a pathway where A goes to B, goes to C, goes to D. That defines the route of flux. But those same processes may be distributed within a population such that the population collectively pushes that flux forward by distributing the functions, or at the community level, where the whole pathway becomes modularized and different members of the community, different cellular agents specialize in one reaction sequence, and the pathway itself is an emergent property of that biological community. As such, we have to think of microbial food webs as complex networks of interactions in which metabolism is distributed and emergent phenomenon. Here we're just looking at an oxygen gradient. The nodes represent taxa scaled to oxygen concentrations, and the edges, the links between those nodes represent correlation. How often do these nodes appear together in a sample? And the colors represent the taxonomic groups. You can see that at high oxygen, we have one set of connectivity patterns. As we move into the lower oxygen regime, we see a change in the connectivity. We see the same taxonomic groups that are able to associate differentially. We have to discover these nested design principles if we're going to harness the power of these microbial communities to develop bioprocesses. And we need to do it now because the world's on fire. And this is a 2009 representation from the Stockholm Resilience Center, in which those green lines in the web represent the safe operating boundaries for the Earth system. We've surpassed in 2009 multiple of those operating boundaries, things from climate change to the nitrogen cycle and biodiversity loss. And if we contemporize this to the present, we've surpassed many more. And so the hope is that we can use that operating system information encoded in the collective microbial system to find solutions that will bring us back into that green in a sustainable way. And the existential problem, of course, in climate change has to do with CO2 emissions. In part, 10 to the 10 gigatons of carbon dioxide emitted on a yearly basis internationally, and we really need to figure out alternative paradigms here. The challenge is that we're addicted to oil, right? We use 1,000 barrels of oil per second, and that drives through this cracking process, the derivatization process, everything that we know in our modern world. From the fuel that drives our engines to the precursors for plastics and pharmaceuticals and pesticides and fertilizers, we are addicted. We need to find another way. And the way that I posit here and I offer is that we find the ability to traverse that prokaryotic plurality, that 10 to the 30, that operating system, traverse that network by defining search functions, by finding microorganisms, engineering them, or finding those native organisms so that we can develop bioprocesses. And obviously, the first order question had to do with biorefining. Can we replace the existing refining framework with microbial bioprocesses that can break down lignocellulosic biomass in defined ways? So can we take the sugar platform out? Can we take the lignin platform out and define like monoaromatic profiles in specific ways? And what we've done over the years in the lab, working with many other people, is we've defined a workflow, a platform technology, where we can take the genomic information, those operating systems encoding in microbial communities from environmental samples. We can take that chunks of code as DNA fragments, put them into E. coli, build DNA libraries, and then screen those libraries for functions of interest. And we've done that for glycoside hydrolases, and we've done that for lignin transforming enzymes. And what's made that possible is through CFI grant that went to the Bioproducts Institute in its early inception to create this biofactorial automation core facility, so that we have this robotic system that allows us to do high throughput screening for those functions of interest. So we can do 10,000 individual wells and plates in one duty cycle. And we have a series of screening paradigms that relate to chromogenic or fluorescent assays, substrate conversion, to biosensors, and more recently selection-based approaches that allow us to interrogate the environmental DNA for functions that we're interested in. And we've had some success over the years working collaboratively, defining enzymes that do the functions, glycoside hydrolases, lignin-integrating enzymes. One great collaboration with the Withers Lab helped start the company Avivo, which is modifying the surfaces of red blood cells, for example. And so there has been technology transfer to come out of this platform. At the same time, we've been able to build an environmental biobank, literally a collection of samples from the environment, aquatic, terrestrial, biofilms hosts that are stored in economical freezers in the Life Sciences Institute. This is a resource for the campus community. We have over 7,000, 100,000 of these clones in libraries that can be used for screening. We have host chassis, biological parts. We have isolate collections and enrichments. And this supports both academic research on a cost recovery basis as well as contract research with industrial partners. So we have a screening facility that does the automation. We have a biobank that holds the samples and integrates that into information management system. But sometimes I feel like we've lost our way. So it took a decade to get to that point where we were actually able to do the screens to look for functions. But where do we go from here? How do we do it? How do we scale? How do we go from just, this is an interesting discovery, this is something curious about the natural world, that's important. But then how do we deal with the world on fire part? And it only works if we do it together. And so this leads to a whole new collaborative paradigm that we had to kind of build as we built the facilities outward. And this brings me to how do we harness the power of microbial communities. Ultimately, we have to harness our own communities to do it properly. So here's a satellite image of the Earth over a year that's measuring chlorophyll fluorescence. So you can see the terrestrial environment and the aquatic environments, the oceanic regime in particular. And what I want you to pay attention to as we move along the equator past South America, you're going to see this pulse of chlorophyll fluorescence. There you go. That's the Earth breathing, literally. That's the phytoplankton blooming in response to some environmental conditions that were optimal for their growth. And so the thought here is, can we emulate, because the scaling is such a problem, the Earth system's already figured that out, right? The Earth system scales the carbon cycle. The Earth system knows how to do it through these phytoplankton assemblages that fix carbon and produce biomass, produce bioproducts that actually structure the food webs that drive carbon transport, which regulates the climate. Certainly we could learn from that scale of process control in coming up with our own solutions to that oil addiction problem. And so we started to think about how do we build photosynthetic cell factories? What do we need? And one of the real rate-limiting steps, it's still a rate-limiting step here at UBC, is we don't have access to the photobioreactors that we need in order to move from the laboratory system into the scale-up production for the proper techno-economic assessments. But if we could look for essentially bioproduction paradigm, not biorefining, but bioproduction paradigms for carbon capture, bioproducts, biomaterials, bioremediation, et cetera. And one of the things that we have been able to do over the years is to isolate a local strain of filamentous cyanobacteria called Sotalinema yahuli that forms these very dense biofilms that has quite a bit of biological and biotechnological potential. It grows like this, though, and so we can't do traditional molecular genetics. It doesn't like to form colonies. It just likes to swim all over the plate. And so that actually led us to do some innovation on our own where we developed hardware. We developed a high-throughput lighting system with LED arrays that allows us to use the automation facility that we have in the Life Sciences Institute to screen photosynthetic microorganisms in high throughput. So this is a technical innovation that now allows us to do things we couldn't do before. Specifically, you're seeing the Sotalinema yahuli grown in a 384 well plate under different conditions. And you can see in some wells it's not growing well, in some wells it's growing very well. We can actually do screening in high throughput for traits that we are interested in. So that's integrated in the core facility, so you can use it if you're interested. And we can do these massive growth media optimization experiments. We don't even have to do any genetic engineering. We can just start to ask, if we change one parameter in the growth media, how does that affect the growth? We can do multifactorial experiments to look for response surfaces that allow us to optimize the growth of the strains. And we've done this with a variety of cyanobacteria, and we can consistently see by just modifying the growth in this high throughput format, we can get yields that are 53, 61, 38, 36% higher. Now this matters if you want to scale a bioprocess, right? Having a growth yield of 62% increased over the norm is significant from an economic perspective. We've moved on to the phytoplankton. We can actually grow diatoms. We can grow coccolithophores in this format. And again, this opens up a whole new possibility of not just modeling the way the ocean works, but bringing the organisms that drive that fluorescent bloom across the equator into the laboratory setting to develop bioprocesses accordingly. We've been exploring other modalities for platform innovation, cell surface display, and 3D hydrogels, some of which you've seen around here, printed with mushrooms. And what this really does is it comes together in a new paradigm for us together at UBC to focus on engineered living materials as a solution, not just a placebo, but a solution to some of the problems that we're facing. Here we can actually generate systems with rational design that emulate the microbial networks for sustainability, for resilience, that actually do functions of interest. Bioprocesses, they can remove waste from waste streams, they can break down organic matter, they can convert it accordingly. And so I just want to end on how do we actually achieve those goals in the near term? Like, what do we need to do collectively? And I think the BPI is an great example of how we can bring diverse researchers together around some common vision. And this is really an extension of the ecosystem that the BPI has already created. Okay, what it does, though, is it brings other units together as well. And so we have a core facility. If I take the gears in the engine, we have this core facility that does automation, screening. It's open to everyone. You can use it. You can innovate with it. We have an environmental biobank where samples are stored and resources, genetic resources are located that can drive that discovery process. We continue to look for CFI support to expand our capabilities. And we have these vibrant institutional partnerships with the BPI, with the Bradshaw Research Institute for Minerals and Mining, with the Life Sciences Institute, where we are right now assembled. But more importantly, we have people like Nick Lin who are driving the community assembly component of this enterprise. So Nick is a postdoc who's supervised by myself and Joe Damon in biogenic architecture in Sala. And he's been helping put together a research cluster that is actually taking advantage of these gears collectively. And so here's just a snapshot picture of the people involved. We want to grow this community. This is the M-Cells Research Collective, if you will, interested in engineering living materials. And this is our workflow, right? We're taking advantage of those gears from the environmental sample collection all the way through to the omics that are supported through the core facilities in the LSI. We can do design, build, test, learn cycles using our automation facilities and design and experiment software. And ultimately, we can fix on specific bioprocesses of interest. And those three current processes relate to the production of omega-3 fatty acids for the aquaculture industry, microbial-induced carbonate precipitation with people like Sue Baldwin in mining and chemical engineering, and engineered living materials, where we're actually building these hybrid structures with mycelium, other microorganisms that have been engineered to do specific tasks. And that would be like collaborations with Joe Damon and Orlando and others. So this is how we're taking advantage of the collective possibility that we have built over many years and the resources at our disposal at UBC. And so in conclusion, what I've told you essentially is that we have to do something about the fire. And one possibility is we can look back into the history of the Earth system and take advantage of the fact that there are 10 to the 30th microorganisms that have thrived for 3.5 billion years, not alone, but in community. And the solution to our problems scientifically is to learn from that community paradigm to engineer new bioprocesses, some of which we haven't even imagined yet. And I will leave you with that note.